Hello, friends. Very good morning to everybody. We are having today's lecture on the subject that is the recessive epistatis. In previous video, we have studied the epistatis, that is the gene expression and the gene which inhibits the characters of the other one, which doesn't allow. And in that, there are two types of epistatis, that is the dominant one and the another one is the recessive one. Whereas, speaking about the dominant one, the dominant allele at one locus or a location which prevent or inhibit expression, the expression of one or more alleles at the another location. It might be of homozygous or it might be of heterozygous. This is dominant epistatis. When studying the summer squash example regarding the color code or fruit color, what we come to know that there are three types of the color phenotypically. But when we take the white one and green one and allow them to fertilize, what we get that the white one completely different. Means what? The dominant epistatis, in dominant epistats or in dominant gene expression, what happens here? The green one was suppressed. What we get? That white one. In F1 generation, it is allowed to undergo self-crossing, white into white, and what we get in F2 generation, we got that phenotypic ratio. We are not talking about the genotypic. We got the phenotypic, that is 12-1 white. His dominant one was more, then 3-1 yellow, completely different. This was not present over in this, and the last one, that is the green one. Here is a typological mistake, typing mistake. It has shown white one, but when you look out at this Punnett square, you come to know that in the last box of the last column, you see the green one, and rest of 12 are of white one and 3 are of yellow one. I must show you this yellow one. These three boxes you are observing, these are of yellow one and the last one is of a green one. Rest of all the among the 16, 12 are white one. So this is what we see, that we got the dominant character in the summer squash. Though the phenotypic coat color of the very upper part, color of the fruit, that is three types we see. There are green one, yellow one and white one. But of course in the ratio, the white one is dominant than the yellow one and yellow one is dominant than the green one. So this is what about the dominant epistatis. So what happens here? Prevention of expression of one or more alleles at another location. Something is inhibiting here. Now what about the recessive epistatis? When we look out at the phenomenon of the recessive epistatis, here though we find prevention of the expression of the gene by recessive non-allelic gene. Non-allelic gene. Though here is also prevention, But what prevents here to express the gene, that is the recessive non-allelic gene, prevents to express the gene, that is the recessive epistasis. Well, while talking about this example of the mice color coat, so look out at the phenotypic for X1, that is the black coat color, which is crossing over the albino coat color. While talking about the genotypes, we find the capital M, capital A, with concern with the albino, that smaller M and capital A. What we get in F1 generation, completely different. Agouti. Look out here. What we find here white one, that is dominant one. But what we find here, we don't find any black coat color, we don't find any albino, we find just any third one. Neither from the parent, neither from the mother, neither from the father. We got the completely different, that is the agouti. Now allow it self crossing, self pollination in F1 generation, which undergoes, and now the agouti shows the genotype, that is capital M, smaller m, capital A, smaller a, which is crossed by another one, on the other hand the another agouti. What we find in F2 generation, look out at this one is the female one, this one is the male one. From the right hand, from the upper side, the genes, and from the left hand side of this Punnett square, we find the male one. So while crossing over it, what we find? We find the 9 are of agouti, 3 are of black, and 4 are of albino. So obviously this is the recessive epistasis. Why it is recessive epistasis? Because it shows the character which were recessive in somewhat previous generation were expressed here, but though the pure parents were black coat color and the albino coat color, the pure parents, and they found the agouti in the F1 generation, and after this F1 generation going in the self crossing or self pollination, what we see that the ratio is 9:3:4. Whereas in dominant, what we saw that the 12:3:1, means whatever this white one was shown over here. This is the white one, and of course this white dominant was seen, and this dominant species, that is the got the white one, and of course this the 12:3:1 ratio. Well, about the recessive epistasis, in case of the recessive epistasis, we saw that the black coat color into the albino coat color, what we got in F1 generation was the August2. Here also we find the prevention of the expression of the gene by recessive non-allelic gene is known as the recessive epistasis. Well, its names only suggest there is somewhat the prevention of the expression of the gene's character by the recessive non-allelic gene. So what we get later on here, that in the case of recessive, the agouti color was found in 9, then 3, that is black one, and 4, that is the albino one. Now we move towards the topic, that is the duplicating factor. What is the duplicating factor and what exact the phenomenon says? Single character is controlled by two independent gene express a seed character. whether the other gene is present or absent. Look out at this phenomenon. A single character in this phenomenon is controlled by two independent genes. Express a seed character whether the other gene is present or absent. So in this duplicating factor, we can find out whether the gene is present or absent, which in the seed character. Okay, so it was observed in the shepherd purse, that is the phenotype triangular seed into top seed shape. These are the shapes of the seeds, and the genotypes, as usual, you are observing on this slide, that is capital double T and capital double D, whereas in the top seed we find the smaller double T and the smaller double D. Well, while we find out its gametes, we see the gametes, that is the capital T, capital D, that is single one, whereas in the top sheet, that is the smaller letters, though is single one. In F1 generation, what we get, that is the triangular seed shape in F1 generation. Means the dominance is somewhere is shown in F1 generation itself. Now allow the generation to the self-pollinate. What we see, the phenotype and the genotype, the triangular shape seed is crossed over the triangular shape. Now the gametes, what we find. So look out at the phenotypic ratio. 15:1. So what type of ratio is this? 15 triangular shape, we find the seeds, and one is of only top shaped. So we completely dominant, but only one top shaped. So with this, what happened? A seed character with the other gene is present or absent. A single character is controlled by two independent genes. So this character of the triangular shape is controlled by two independent gene, and which is expressed. So by looking at this, we come to know that there the triangular shape we found 15 in number, whereas the top shape only 1 in number. Well, when we look out at the genotype of this, we can clearly see that how differ there is the changes in the genes and the what we can say, though the character express, but there are some slight changes in the genotypes of this. You can look out at this Punnett square, and you may find out that how this is the difference made. So this is what about the duplicating factor. Well, we have to also remember and remind these factors and the ratios of different genes interaction, what type of genes interactions are seen in this hybridization, or in this topic. What we see, there are different types of ratio of every experiment of the done by the scientist, whereas monohybrid, dihybrid, then we see the gene interactions. Into that, what we find, the complementary gene, allelic, non-allelic. In case of allelic gene interaction, we find incomplete dominance in Mirabella jalapa. We find the ratio, that is 1:2:1. Next on, that is the codominance. What is the codominance? Coat color of the cattles. So in codominance, what we find, that the red and white, the ratio, that is the one red, two roan, and one white, means 1 is to 2 is to 1 ratio. In allelic gene interaction, we find 1 is to 2 is to 1 in case of incomplete dominance as well as the co-dominance. Whereas non-allelic gene interaction, what we see, complementary gene interaction, epistasis, duplicating factor, supplementary gene actions. Well, while studying this complementary gene action, we got the ratio that is the 9 is to 7. Somewhat the in case of epistat, there are two types of epistasis, dominant as well as the recessive. In dominant epistas, we studied the summer squash and we find the 12 is to 3 is to 1 ratio of the dominancy. Whereas in recessive, we find the little bit different ratio, that is 9 is to 3 is to 4, that is 9 agouti, 3 black and 4 albino. So friends, this was all about the gene interaction what we saw and studied. Rest of the part we'll be going to study in our next lecture. Thank you.